Strength in Leos.
Welcome back, guys, to the Strength in Leos podcast. We have another awesome guest with you guys today, Travis Wyman. Dr. Travis Wyman, how's it going? Welcome to the podcast.
Oh, it's going well, sir. Thank you for having me on.
Of course, yeah. I know you're well respected in the reptile community in general. I know you're respected in the science community as well and have done your rounds of podcasts we were just talking about, so I'm glad to get you on the show today.
Well, hope to have a good conversation here and hope everybody enjoys what we have to talk about.
Definitely, yeah. So for people who just aren't interested with or who aren't familiar with you and who you are and kind of your background, can you just give people a little bit of background on who you are, a little bit of what you do and that kind of thing?
So my reptile background is probably a lot like most other people's. I got started real young, caught my first snake when I was 5 on the playground at preschool, brought it home, kept it in a little glass tank for a couple days, had to let it go. From that point on, I was pretty much catching snakes and fence lizards and salamanders down at the local pond and stuff for years and years and years and years. I made my first real snake purchase when I was 14. Bought a corn snake at a pet shop down by my father's house. That snake was with me for 25 years.
Wow.
So, he moved with me all over the country and made it through graduations and marriages and divorces and birth of my child. And
Wow.
I have expanded outward since then. I've got a fairly eclectic snake collection. Mostly I do have one set of lizards, although they're really snaky lizards, so still can't be accused of having anything with legs.
Not yet.
Not yet. Well, my kids got crested geckos, but those are her crested geckos, so
Not yours.
I don't have anything with legs. I professionally, I am a research scientist in the field of forensic metagenomics, which is a really complicated way of saying that I do a lot of work in the field of using genomic and next generation sequencing and molecular techniques in a sort of forensic capacity to identify samples and things like that.
Nice. Cool. So when it comes to your academic career, how did you go about figuring out what you wanted to do, being interested in reptiles growing up, and then kind of using genetics in that field of reptiles also to combine your passions?
I was obviously catching snakes and stuff at a really early age. I was always into biology. So I fell into biology really, really easy with life. And as I worked my way through, elementary school, junior high, high school and stuff, I knew that I wanted to pursue something in biology. And when I got into college, I started along the course of a major in biology, but then I just the more I looked at the field itself and what I could do with it, my brain started kind of moving in the direction of smaller and smaller and smaller, figuring that, it's a lot easier if you're at, I don't want to say the lowest level, but at the most basal level of biology and the understanding of biology, because it's easy to go up steps. It's a lot easier to be a microbiologist or a geneticist or whatever. And if that proves not to be where you want to be, to step up and be just a biologist or just a zoologist or just a field biologist or something. It's a lot harder to pursue just a career in biology and then get everything done in biology and be like, but I want to be, much, much further down on that basal level to try to, make my life and my career. So when I had that realization in college, I shifted my major from just being strictly biology to being microbiology. And then I took a year between college and graduate school off to just do basic research, mostly because I had some friends that were a year ahead of me. All of them went into graduate programs and all of them scrubbed out. So there's a part of me that was just like, maybe there's something about just basic research that I don't understand and I should figure it out. I went into basic research. It was studying transfusion related acute lung injury, which has nothing to do with microbiology, but it was still, again, that basic molecular and cellular level of stuff. Really enjoyed what I was doing. So then applied to graduate schools, went to Emory University and got into the genetics, the microbiology and molecular genetics program there, which is what I got my doctorate in. And then, again, that's that very basal level of biology. If you understand genetics, then you understand how genes interact. You understand from how genes interact to how proteins interact to make how cells interact, how tissues interact, tissues to organs, organs to animals, you can work your way up that tree. And it was just, for me, it was just a field of study that managed to really engage me. And so I just kept pursuing it to, where I ended up now, where you just use, like I said, in the research capacity, how can I identify what's in this sample using what DNA I can tease out or molecular markers that I can tease out, things like that. As far as it applies to my interest in the hobby and stuff, it gives me a very unique and deeper, broader level of understanding to how, genetics works with how morphs work and things like that. It's one thing to say, an albino is a lack of melanin. I can say, a T minus albino is lacking the enzyme tyrosinase, which is the most fundamental enzyme in the initiation of pathway to cause melanin synthesis. So if you pull that pathway apart at the very beginning, that's why you get an animal that has no melanin. But by contrast, or by comparison, a T positive type has still got that enzyme. So it's beginning to produce some degree of melanin along that pathway, and somewhere else the process gets broken, whether that's, in the melanin synthesis that gets broken, or the transport of melanin that gets broken, or the stability of the melanin that gets broken. So I can have these much deeper understandings than just, the snake doesn't have black pigment, so that's why it's yellow and white, which is how most of the general public would see it. Now, people in the hobby tend to understand it a little bit better. But a lot of the reason that people in the hobby tend to understand it a little bit better is because of people like, myself, Warren Booth, Zach Loafman, Nick Mutton, Justin Julander, other guys like that, where we have that deeper understanding and we want to convey the deeper understanding out to everybody so that we can, help get them to understand it on a more fundamental level than just snake with funny color makes pretty.
Right, yeah. That's what you see most in the hobby. Like, I think you might have just screwed a few people's brains over explaining that, because they just know kind of the basics. But it's really awesome also how you can understand things from the micro level of biology, but then also going to a more macro view and really understanding the different levels it is to genetics, because I think it's a lot deeper than, as you've noticed, probably as people in the hobby compare things also to the scientific community and really getting down the definitions and also the processes of how these different interactions happen. Yeah. So when it comes to just genetics in general, you probably have seen just the reptile hobby and how it's evolved from, people just slapping names like hypo on things that aren't really displaying what that word actually means in the scientific community and things like that. So how do you think the evolution of genetics and understanding genetics from the scientific community also just transferring to the hobby and getting the hobby on the right path as well?
I think that it's sort of a weird tug of war type of situation. There is a lot of inbuilt reticence and resistance in the hobby. part of that I think just comes from the nature of who reptile keepers are. We're already viewed as being kind of fringe or not normal because compared to cats and dogs and birds and goldfish, snakes and lizards and frogs and toads and things are seen as weird and different and sort of counterculture,
Right. Yeah.
going against the grain. And so the hobby itself has sort of the same kind of mentality when it comes to understanding genetics, understanding proper terminology and things like that. It's very much, that against, well, I'm just going to go against the grain. I don't care if there's a reasonable explanation or something. This is how I've decided to view it. And so I'm just going to view it that way. And that kind of stuff frustrates me. And, that's why I try very hard to, not just explain when something is mistaken or wrong, but why it is that it's wrong and how looking at it in the right way can actually both further the hobby in terms of just understanding, morphs and how they work and how to, make different combinations and things, but also it works towards just being more scientifically literate and more literate of how you then have to deal with your animals and look at your animals if you're trying to do other things like, if you're working with a new species and you do some research into it and you end up reading some scientific papers, not just taking and vomiting back the most basics of that scientific paper, but understanding the critical process that goes behind what that paper is saying and why some of the stuff in there may be very important and some of the stuff in there might be a little suspect. And so you have to dig deeper than what's just in that one paper. So I have seen a lot in terms of both the pushback from that counterculture mentality, but at the same time, there's a lot of people that, I guess, sort of a newer generation of people that aren't quite as caught up in, in the old school way of thinking and I don't mean this in a derogatory way, but like the old idols and this person said it and this person is one of the old people from the hobby, so they have to be right. They're interested in hearing a bit more why there's disagreements or what this information is and where it's coming from rather than just swallowing it whole. So there's a bit of a thirst for knowledge that I'm seeing now that I really enjoy and appreciate that, for a while I think was lacking.
Definitely, yeah. And it's really cool to see that progress. And you kind of touched on my next question a little bit more, but in terms of that competency when it comes to reading the literature out there that exists upon genetics, I think you've probably heard more than anybody else the incomplete dominant, the super form and things like that, and co-dominant, that whole debate that people go on. So like, why do you think that competency matters in using the correct terms when it comes from what you see in the literature every day in the field and then transferring that over to reptile genetics?
It's a difficult
Opening a can
question
of
to
worms
answer.
here. Yeah.
Yeah. I mean, on some levels, it's insignificant. If you are calling it one thing, if you're calling it co-dominant, but it's really incomplete dominant, why does it matter? You know what you're talking about. Okay. Yeah. You know what you're talking about. And yeah, the other people in the hobby know what you're talking about. But to me, why it matters is, in this connected world that we live in this time where we're having to go up against groups that are trying to shut us down. We've got USARC fighting on our side and stuff. We need to have some degree of literacy so that we have legitimacy. And that can mean that if we're talking about things that are just completely scientifically inaccurate, we look a little bit hypocritical then when we have to issue a challenge back to something like, the paper, a decade or so ago when they published that paper saying that, Burmese pythons were going to be able to go all the way up to the coast of Connecticut and all the way over into Colorado and stuff. Like, scientifically, that's just not a viable paper. And it's one thing for us to say that and then also be using correct scientific terms and scientific sources in literature and showing our literacy and understanding in that regard, because then it's, this is people who understand science basically pointing out scientific flaws in a scientific paper versus these people who don't know what they're talking about, who use their own made up gobbledygook language and terminology telling us that scientists don't know what they're talking about. It's hard to reconcile that when it's just, no, you're wrong, but I'm using all these terms wrong, but you have to listen to me because I'm right. It's a hard pill to swallow when you're going up against legislators and things like that, because when they hear that you don't know what you're talking about, then they just kind of dismiss everything. And some people say that's an extreme view because, why do the legislators care about whether or not you're using genetics terms correctly if you're arguing about the environmental impact of Burmese pythons? Well, yeah, it may not make a difference on the one hand whether or not you understand that tyrosinase is an important enzyme for the synthesis of melanin, but if they see that you can talk with that scientific literacy, they may never have that conversation with you about the Burmese python and the color patterning. But if they see and they hear that you're already using this legitimate scientific terminology, then when you challenge them and you start arguing counter science, basically, or opposing science or pointing out flaws in science and scientific publications, it at least puts you on a more level playing field.
Yeah, definitely. And I think it's really important that we just as the reptile hobby that we have that credibility, right? I think when we talk sometimes and you know what happens on social media and then things happen with drama and things like that, but also when it comes to just the credibility of the hobby in general, when we know what we're talking about and using the correct terminology for some of these genetic things that are happening in there, I think it brings a lot of credibility to the hobby. And then also that we just really care about the intricacies and the passion of the reptiles in general. And it's really cool when we are able to get that knowledge and then also spread that knowledge to other people to establish that important credibility that we have.
It's like credibility is a great word for it. It's a credibility factor. If we're credible, it's great if we can all have a conversation amongst ourselves.
Yeah.
Because we all know what we're talking about. But it's even better if we can have a conversation not just with ourselves, but ignore legislators and the animal rights groups and stuff that are coming against us. If we're trying to get support from other communities, or if we just want to begin to engage other communities, it's a lot easier to be able to have these discussions with, people that raise birds.
Right.
Dog breeders, for crying out loud.
Yeah.
If you're both having the same conversation, it's a lot easier rather than if I'm just using bastard hobby terms that only we as reptile breeders understand.
Yeah.
And then the bird people and the dog people are like, I don't know what you're talking about, so I can't engage you. And they may be really interested in engaging us. They may not be, but they might be. And so finding that common ground when you're all coming in at the same level gives you that credibility, but it also gives you, like I said, that common ground, that shared level with each other.
Right. Especially when we're talking about genetics and some of these same things that we see within the reptile community, even across reptiles, if we're talking about snakes versus lizards, but even when you're talking outside of reptiles and we're bringing in like birds and fish and things like that, we could have this same vocabulary that we're using to describe similar phenomena that are happening within different sectors of just exotic animals, which is also super cool to think about.
Yes. And that also just goes to, again, just literacy within your own group.
Right.
I, for years and still now, like I will hear people talk about like ball python genetics as if the genetics of ball pythons is completely separate and different and unique to any other animal or organism on the planet. Like,
Right.
genetics is genetics is genetics.
Yeah. At the end of the day.
It's what it is. Yeah. At the end of the day. And, I can say that really simply by saying, like, once upon a time, I got in a forum argument with somebody over whether or not an albino Burmese and an albino ball python, if crossed together, would make an albino. And the simple truth is, now assuming we're talking about a T negative albino amongst both of them, they're absolutely compatible because that's the nature. There's only one gene for tyrosinase. If I'm missing it in my ball python and you're missing it in your Burmese python, then by absolute 100% understanding, the hybrid that you make from that would be an albino.
Yeah, by definition.
Right, by definition. But it goes beyond that. Like, tyrosinase negative people are albinos.
Right, yeah.
It's the same gene. It's the same gene in mice. And we know for 100% certain that it's the same gene in mice because they have knocked the gene out in mice and then spliced the human gene into the mice and turned albino mice back to normal colored mice. Like, it is the same gene.
Right.
And, you see it with dogs, with cats, with horses, with whales, there are albino strains of mold. There's mold that synthesizes melanin. If they knock that gene out, instead of being a brown mold, it's a white mold.
It's crazy.
That gene is carried, through evolutionary history. And understanding that means that you can have a better understanding of how a lot of these genes work. By extension, you look at the spider ball python and the jaguar carpet python. Both have reduced patterning, both have neurologic issues. It's probably not a far step to say, look, these both are behaving very, very similarly. Maybe this gene is the same, and having a more fundamental understanding of how that gene is the same, then you can look more broadly and be like, well, where do we see something that reduces pattern and also has this neurological issue? And maybe we can actually find out the absolute identity of that gene. We don't know what gene it is that gives you the spider ball mutation morph. But, if you look more broadly out there, are there dogs or cats or mice or something that have this sort of weird pattern to them and a weird neurologic balance type issue where we have identified the gene? And then can we look at that gene and go, okay, I know it's this gene. Now I can look specifically for that gene in the snakes that we're dealing with and see if it's the same gene that's been mutated.
Right. Yeah, and I think it's really interesting in reptiles when we name certain things and when we talk about genes versus morphs kind of thing. And when you're talking about the gene that actually makes that change happening and is making that phenotype have a certain kind of look, when we're looking at that thing that's happening, we could also apply it to different species. And people think, well, oh no, it's this thing and it has, this name. This is like the X gene or whatever. And it makes this kind of thing. And when we're applying it to different things, we're like, no way that could be the same thing because it's in ball pythons and it does this, when that same effect is also happening across species. And I think people have a hard time connecting those different things together. And when you see that, it's like, I don't know, you just question people and their ideologies and how they're thinking about that kind of thing. But when it comes to just literacy and competency and genetics and things like that, obviously you doing podcasts like this help get that word out there and spreading that. But also just within the reptile community, how can we start these conversations and spread this information in a way that's, easy for the layman to digest and really get a grasp on, but also be able to do further research into learning how genetics work from the basic steps of Mendelian genetics to, get correct information out there for people to understand?
I guess the first thing is saying becoming scientifically literate it sounds kind of like a trait or, just, well, of course, become scientifically literate.
Yeah.
But it's part of being scientifically literate means that you stop and you question even the answers that you're given from apparently authoritative sources. So if you hear about a paper talking about an animal and a specific behavior, and you read that paper, you don't just stop at reading that paper and decide, okay, I've read a paper about this behavior. That's the absolute, complete, total sum knowledge that there is on that. You need to think, okay, I've read this paper about this behavior. I need to look at this from sort of an opposite manner. What if I wanted to challenge what this paper was saying? What would I look at in this paper that might make me think that this paper isn't an absolute? Well, if you're dealing with a species that has a range that's, half of the eastern United States, but they only looked at a population around Miami, let's say, then that would be a thing that would make you give pause. Be like, okay, but if they only looked at, 100 snakes that they caught over 3 years around Miami, then how can we say that those 100 snakes are representative of every single snake across the entire range of the eastern US?
Right.
So that's a question to ask, and apply questions like that, and then go looking for more answers or answers to those questions. Okay, so I read this one paper about this population in Miami. Has anybody else written papers about the populations in South Carolina, in Virginia, in Massachusetts? Read all of those papers and then combine the conclusions from each of them and see if out of that sort of combination in your head, there's a bigger picture that's built up. And now that bigger picture may be, yeah, what they saw in Miami is the same as what they see in South Carolina, is the same as what they see in Virginia, is the same as what they see in Massachusetts. But what it might be is Miami snakes behave this way, South Carolina snakes behave this way, Virginia snakes behave this way, Massachusetts snakes behave this way. So you're seeing a range of behaviors. And that means that if you're going to work with that snake, that species, you might need to take into account what the founding stock for the animals that you're getting are, because that could determine how you have to treat that snake in your care. Being able to read broadly like that, asking questions. In terms of where to get, the ideas and understandings of just basic genetics, I would say go buy a textbook, but we all know textbooks are pretty much become an outdated thing these days.
Right, yeah.
But you can find all this stuff on the internet. But again, that's where critical thinking and critical understanding comes in too.
And a bit of common sense as well.
Right, a bit of common sense. Like you can Google, basic genetics, but are you getting basic genetics from Bob's Discount Basic Genetics Barn as the first, hit on Google because Bob decided to pay to be the first hit on Google? Or is it coming from an actual university that just has an intro to genetics course free online? Or it's the slideshow for a basic genetics course presentation from 2 years ago that they don't care about, being posted online. Or, there are a lot of even online learning courses through community colleges and things, or just trying to remember the one my father has. I think it's called Great Courses or something. You pay $15 and you get the module that you're interested in, or if you subscribe for $15 a month and you can go into different classes basically. So get that information from reputable sources and use it to build up your knowledge base to then inform that literacy that I said you should have, because that gives you a better ability to question both what it is you are reading and understand what it is you're reading. It's also really important to recognize there are people out there like me, like Nick Mutton, like Zach Loafman, Justin Julander, Ben Morrill, we like talking to people as long as they're willing to have intelligent conversations with us. Engage those people. I don't mind when people come to me with questions. If you have questions, ask me. If you found a paper and it seemed interesting and you liked it, but there were parts of it that you didn't understand, just be like, look, I read this paper. Could you help me out here? Now, realize that one, I can't necessarily just drop everything and help you right at that second, but, I'll try and read over the paper. I'll look at the part that you're having difficulty with, and I'll help you out. I had a buddy who he had this whole paper on this bizarre species that he's working with that like nobody really works with. And he read the paper, he was all into it, and I could tell that he was all into it, but he's like, I have no idea what any of this means, especially this. It's like, oh, okay, I get that, because there's a lot of jargon there. Let me break that jargon down for you. And this means this means this. Do you understand better now? And his answer was, yeah, that makes a lot more sense. And it was a paper on how to identify these species because there's like 9 or 12 different species, but they all basically look the same. They're just little brown-black snakes. And how do you tell them apart? Well, you have to do like these specific scale counts, and well, what does a derived characteristic mean? Basically, it's a characteristic that only that species has because evolutionarily, either it developed it independently or all the other ones lost it. So, when you have that vocabulary, when you have that mentality, and then when you have the ability to just reach out to somebody and ask them. If you find papers that you like, you can try reaching out to the author of those papers too. A lot of times those guys are willing to talk as well. Again, remember that they have their own lives and they may be busy, so they might not be able to drop everything and get to you, but they will usually get back to you in some capacity or other.
Definitely. Yeah. And I think one thing, even just from doing the podcast, a lot of people who are into research and are publishing research love to get that information out to people who specifically are interested within that research field and want to get that scientific communication out to other people as well. And a great platform. So having platforms like this as well within the reptile community are great ways for people to get this information that they're interested in.
Yes. Another thing I want to throw out, and this, I guess, is sort of a personal gripe, understanding what peer review actually means. I have seen a lot of people in the hobby throwing out how, well, I'm putting out this information, it's been peer reviewed. That's not what peer review is. Having your friends read over what you're telling people is not peer review. The same way, like you're thinking of peer the way they use peer with peer pressure with drugs. That's your friends are trying. Peer review doesn't mean your friends review it. Peer review means other people with a similar degree of understanding have gone over your material and had a chance to critically analyze it. When I had to write papers and stuff in research in graduate school, and my peer review and my papers would go out for peer review, it wasn't going to the guy in the lab next to me who knows me. It was going out to other colleges, other graduate schools, other research centers, guys that I may not even know. And they're the ones reviewing it and coming back to me. And in some ways, those people who are doing those reviews can be rather harsh and brutal, not because they don't like you, but because they're literally doing what I said, that whole part before, is they read the paper and they look for the things of how would I pick this apart to understand how it could be, how something important here could be missing. And that's part of that critical thought process. So understand what peer review really means, and also understand it in terms of if you find a paper, not all papers, not all publications, not all journals are created equal. There are some journals where you can pay to have your article published without any kind of peer review. But because they have a really fancy name, people just assume that it's a credible journal, and therefore that means it's legitimate science.
Right. Yeah. And yeah, the peer review process, once you look into like actually how that process goes, they really just like tear your papers apart and like go from like thing to thing and really get into the nitty gritty of those things. But it also like really contributes to that information, and having that reliability of like what that information you're saying is really proven and has really been looking or has been looked deep into that information, which is also just really cool from an understanding aspect to know what you're actually reading is has some reliability to it as well. Before we go on, I want to say thank you to our sponsors. Impeccable Gecko is a small to mid-sized family-operated hobbyist breeder operation. They specialize in leopard geckos with high-quality genetics. Their goal is to provide customers with the absolute best animals that they could produce, coupled with honest and transparent business methods. At Impeccable Gecko, integrity and animal welfare are above all else. Impeccable Gecko is a strong supporter of the Strength and Leos podcast, so follow them on social media and check out what they have available. John of Gecko Boa started early, like most of us, catching lizards and snakes in his backyard at any chance he got. Over the years, he has kept and bred more than 80 species of reptiles. In recent years, his focus has turned to specializing within the genus Eublepharus. He has worked hard to pioneer some of the most cutting-edge leopard geckos while maintaining genetic purity and honesty. He also keeps the most diverse collection of pure Eublepharus species in the world. He has coordinated with biologists around the world and has contributed DNA sampling and distribution range info to help sort out the cladistic relationships within the genus. Check out geckoboa.com for more info and available geckos. Suburban Geckos is operated by Chris Charlton. Chris's passion and enjoyment for herpetoculture, and more specifically leopard geckos, drove the desire to take his hobby to the next level. With experience in breeding and raising reptiles well over 20 years, Chris is dedicated to producing quality animals for both the hobbyist and breeder alike. Suburban Geckos treats each animal with the utmost care and respect, and cuts no corners when it comes to health and integrity of their geckos. What started off as just a couple aquariums has grown into quite the collection in just a few years, and they wouldn't have it any other way. The dedication to their animals is unparalleled and evident in the quality and health of their animals that they produce. Exemplary customer service is another area where they excel. They're readily available to answer any questions you may have regarding their animals, husbandry, and etc. For more info and to see their availability, check out suburbangeckos.com. Blizzy Bold Leos, while new to the scene, is taking strides in following the footsteps of top-tier breeders and keepers in care and ethics while aspiring to bring fresh looks and crosses to explore latent potential. Prior to becoming a breeder, Greg of Blizzy Bold Leos completed hours of extensive research on lines, ethics, morphs, and how certain traits interact with others in order to begin projects which are coming to fruition already in the second season of breeding. With a major focus on line breeding the correct way and customer service done right, Blizzy Bold Leos aspires to be a breath of fresh air to the customers and keepers they encounter. Be sure to check out their Instagram, Blizzy Bold Leos, and stay up to date on project development, availability, or just tips on having healthy animals in your care. Dab Exotics is a hobbyist breeding operation that was started in 2017. They specialize in producing healthy leopard geckos from top quality genetics. Dab Exotics is an awesome supporter of the Strength and Leos podcast, so follow them on social media and check out what they have available for sale. Shane Kelly is a blue-collar, working-class guy that has a reptile breeding business with his wife and kids. They are currently working with high-quality ball python morphs, western hognose snakes, and select leopard geckos with a focus on bold stripes, and they're looking forward to adding Albi, Snow, and Ghost this year. They have been listening to the podcast since the beginning and are proud to be a Season 4 sponsor. Go check out their social media, Instagram, Facebook, and YouTube, and also check out their morph market to see what they have available. So when it comes to people in the reptile hobby, we kind of talked about like information, getting that information out there as well. I think a lot of the big things that we see in terms of genetics and misunderstanding comes from discussions in the reptile community. So what can people like you who do research, who communicate with other scientists, the daily really communicating your research, what you're interested in, and then also just getting ideas and bouncing ideas off in a lab. What are those things that you look for in discourse and getting information across to different people that is a productive way? Because obviously scientists are just as passionate as reptile keepers are with their research and things like that. So how do passionate people have a conversation? It seems impossible these days. So what tips can you give us?
So the first thing I would say is, if you're really interested in having a conversation and a discourse with somebody, don't go in as if you're right. Go in as if you're willing to learn. Recognize that if people are asking you questions or challenging your viewpoint, it is not necessarily because they're saying you're wrong. It's possibly because they, one, don't know what you have already looked at. And so they're trying to establish a baseline so they know where the conversation can start, what commonalities you have to then continue that conversation on. And if you go in willing to learn and having an open mind, I mean, that's absolutely necessary to have a conversation or a discourse. You can't have a conversation with somebody who's already made up their mind and made a final decision on something.
Right.
That's not having a conversation. That's just looking for justification or affirmation of the point of view that you already have. Recognize that not everybody communicates the same way. So what may seem blatantly, flagrantly obvious to you, and what you may think you are explaining in a clear and concise manner, may not be as clear and concise to the person you're talking to. It may not be blatantly obvious to the person you're talking to. I will double down with that on social media, Facebook, Instagram, forums, things like that. The typed out word, without being able to hear somebody's inflection, without being able to see their face and stuff, it's really bad at conveying the context of information. If I ask you a question face to face, you can hear in my tone of voice whether I'm asking because I'm interested, whether I'm asking because I'm doubting your credibility, whether I'm asking because I'm confused. If I just ask you that question online, you can interpret that any way you want. And if you interpret that the wrong way and then you come off on the wrong foot, now we're fighting.
Yeah, because there's no body language. You can't see any of that expression.
Right. So try to be as open-minded and as non-reactive there as possible. And there are some forums and things where, if threads are going and they're going crazy, moderators can hit the slow down button and keep you from posting more than once an hour. You as a human have control of yourself in that regard too. If you read something and it gets you angry, you can type up the response, but maybe hold off on sending that response for half an hour or something, and then go back and reread, once you've cooled down, both what was said and that got you there and what you've written, and see if maybe when you read it at first, there was something else going on. On your commute in, you got cut off and you were already in a bad mood and you had a flat tire. And so when you read this post, you got angry, not so much because of the post, but because of all the crap that had happened to you earlier in the day. And it just spilled out in that moment. And then you typed out an angry reply. But when you go back and reread what was written, you can recognize, now that I've calmed down, maybe I made a mistake here in what was being said. So I can change what I was going to post. You can still disagree with the person if that's where you're at, but you could tone that down a little and maybe be like, look, I disagree with you, but I disagree with you for this reason here. Try to express yourself as fully as you can, as respectfully as you can. I know that can be hard. I even fall victim to it myself sometimes.
Yeah.
It can be real difficult. And sometimes, we all get tired of just hearing and seeing the way it is. When I have to go up against somebody who's just obstinate about them being right and not willing to listen to anything I say, after 3 or 4 go rounds with that person, my frustrations will get the better of me sometimes. I'm not going to lie. And I am sure that it comes across in the way that I type things. But, if I initially engage you and say, look, I disagree with you, and this is why I disagree with you, I'm not saying that I disagree with you to be a jerk. I'm saying that I disagree with you because I have my reasons. It could be real simple, it could be real off the wall, but we can have that conversation only if we agree that we're going to have a conversation.
Yeah. And I think that's the most important thing is like, are you really going into that conversation being a little bit humbled to the fact that you may be wrong and what you're saying, especially when you're talking to certain kinds of people, certain kinds of personalities. I know in the reptile hobby, it's a big group of people with big passion, big heart for reptiles, but also some interesting people that we see in the hobby. So it's really important to also look at like, what are you talking about? Even like just knowing what hill to die on kind of thing is also super important. There's some like tiny like things that haven't been proven for fact in the scientific community yet, and there are small nuances where you can have that debate. But is it being constructive? Is it, are you showing both sides, or are you just going into it strong-headed and just wanting your opinion to be out there? So that's also a really important thing that I think people sometimes they just don't care about. They just want to talk and they want to be right, which is the biggest thing I see
Yeah.
so often.
And and that's your comment about having an opinion is a good one. An opinion is kind of like a feeling. it it belongs to you, but that does not make it fact. It does not give it the same weight as somebody else's opinion. I could pick up a cat and say, this is a cat. And somebody else could be like, well, in my opinion, that's a dog. And my opinion is just as valid as your opinion. Now,
Yeah.
that's an incredibly simplistic way of looking at it, but it's making a point. No, I'm sorry, your opinion is completely invalid there. Just because you have an opinion doesn't mean you're right to have that opinion. Objectively, you are wrong. There's no ifs, no ands, no buts. Now,
You have to put that on a t-shirt. So, I like
now,
that.
there's obviously a lot more nuance in some of these conversations that we're having. Like the way hypo is used in the reptile community.
Mm-hmm.
What most people use for hypomelanistic in snakes is not hypomelanism; it's hypopigmentation.
Mm.
Because all of the pigments are toned down in those animals. You look at
Yeah.
the hypo ball python. That's not a hypomelanistic ball python. It's a hypopigmented ball python. The flip side, the things that really are hypomelanistic, people don't recognize those things. So like lavender albino ball pythons, caramel ball pythons, ultramel ball pythons, those are hypermelanistic. They have less than the normal amount of melanin. So that's what hypo is, the prefix for less than, and melanism. So telling people that, well, no, your hypo ball python isn't really a hypomelanistic ball python, it's a hypopigmented ball python. Again, that goes down to that scientific vernacular that they don't understand, but there's a reason I can say that. And, I'm not saying it to you to be condescending. Usually I'm saying it because it's something I can say just even in passing. It's a small tidbit of information, and it may be taken by somebody, it may be completely ignored by somebody, but as people begin to hear it and as the communal whole starts to think about it, then it can start to gain traction, and then people can start having those, okay, I've heard Travis say this a couple times. Why? Well, maybe I should just ask Travis why he's saying that. And then we can have that bigger conversation, but recognize, is Travis saying this because it's just his opinion? Well, yeah, in some respects, but my opinion also has this, I can bring the scientific fact in to back me up. I'm not just saying, hypo ball pythons aren't hypomelanistic because I've decided they're not. I can't back that up for you, but I can tell you that they're not, So it's a weird dynamic in how you play off of each other there in those conversations. So recognizing I guess for lack of a better term, just a basal opinion and an educated and informed opinion is an important factor.
Yeah, and I think when we're talking about these feelings, people, just like you had an attachment to your albino corn snake, if like someone came up and told you like that actually wasn't an albino, this is actually what it's called, and this is like the scientific information behind it, you might be like heartbroken, like no way, like this is an albino. I've known this to be an albino my whole life, and someone just flips your whole world upside down. I feel like that happens with genetics a lot of the time in the reptile community, where people find this piece of information and they're like, whoa, this actually isn't a hypo ball python. Like, do I have to change the name of it? Is like I had marketing tied to this and all of these things. And I think these feelings really get attached to some of these terms. And then when it comes to that, people don't want to backtrack, and it gets messy with those kind of things as well.
Yeah.
Which adds another layer to it.
Your comment about backtracking is a really good one. Science, I don't want to say that it backtracks, but it self-corrects, basically. You know, science is not about absolute answers. It's about interpreting the data, explaining the data that you have from the interpretation that you've made, but recognizing that more data could change how you have interpreted that. So, you know, the amel corn. Amel corns, you know, amel is amelanistic, which means without melanin. I hate to be the bearer of bad news, but amel corns are not amel. We know the gene for it, and it is not a gene, it is not a mutation to the tyrosinase gene. Those animals actually do produce melanin at a low level. It's just that melanin is not properly packaged and displayed.
Interesting.
So realistically, scientifically, correctly labeling them, they would be hypomelanistic. They would not be amels. Now, I am not saying that the entirety of the corn snake hobby, from, you know, every corner and every facet and on Morph Market and everything, needs to stop calling them amels. But just knowing that technically they're not really amel, I do think is an important thing. And the reason we called them amels is because traditionally they looked like what we believed an albino animal, you know, an albino, a T-negative albino would look like, before we had the information to understand and to interrogate the genes to show that it wasn't actually the tyrosinase gene that was the mutant gene. You know, we see it with like the boas, the call albino and the sharp albino. Now we know those are incompatible morphs, which means only one of them could truly be a T-negative albino, and the other one's a T-positive albino, even though they both look like they could be amelanistic. So nobody's done the work to find out which one is and which one isn't T-negative. But I can tell you definitively, at least one of them is T-positive. They may both be T-positive for all we know. But at least one of them has to be T-positive, even though they both look like they could be T-negative. So again, that's that literacy, that's that understanding, and that's that self-correction. You know, we're not backtracking on it when we say, okay, I recognize now that amelanistic corn snakes aren't amelanistic. We're going to still keep calling them that because, you know,
that's
the entirety of the hobby, from, you know, little 7-year-old Stevie who's seen the pictures and comes running up to the table and he's like, that's an amel corn, that's the one that I want, all the way up to, you know, Bill Love from way back in whenever, when he was working with corn snakes and brought the first ones in, you know, they've all called them amel. At that point, it's just the accepted nature of it.
Right, yeah.
So we'll keep calling them that, but if we all could admit, we understand that these aren't really amelanistic. That's the progress and the self-correction that's needed. And that happens in the real world with science too. A dirty little secret in science in the terms of paleontology. Technically, Tyrannosaurus rex, according to nomenclature systems, shouldn't be called Tyrannosaurus rex. The first
Whoa.
fossil that was T-Rex and was described was actually found and described as something else before the fossils that were named as T-Rex, and then years and years later, somebody went, so that one that was found like 12 years prior, that's actually T-Rex. But according to the naming convention, the first name that's out there is the one that takes precedent. But the entirety of the paleontology community was like, look, guys, we're not going to stop calling it T-Rex because that's literally what everybody knows it as. So,
Yeah.
But, paleontologists know, okay, so really the type specimen isn't T-Rex. It's this other thing. I wish I could remember it, but that's not one of these things that I've committed to memory.
Yeah. We're just flipping things on people's heads today.
Right. You can, you can have that information, you can know that information, and you can still recognize, we call it T-Rex. Technically we're wrong. We all know that it's really something else, but we're not going to change it because there's just this precedent now, and it would be stupid to try and buck that precedent.
Interesting. Wow. That's crazy information you just gave out there. I wanted to actually ask you something about just albinos in general. So in terms of the different types of, strains of albinism and things like that, I know it varies across species. We talked about, boas, where there's the two type of albinos that aren't compatible with each other. And leopard geckos, we actually have 3 types of albino, none of which are compatible. And then I also know in certain animals like retics, for example, they have different strains of albino which are compatible. So in these situations, what are things that are happening within like this thing that we call albinism and different strains of albinism, if you want to call it that?
So basically, you're looking at that from 2 different directions. With your leopard geckos, it's very much like the ball python or the boa constrictor thing. You have these morphs that look very, very similar to one another, but none of them are compatible. So that means each one of them is a mutation to a different gene that isn't related to the other genes. Now, I told you, tyrosinase is that first gene, first enzyme that starts the whole pathway for melanin synthesis.
Right.
Depending on the organism that you're looking at, obviously more simple organisms have shorter pathways. That pathway could be anything from like 5 steps to like 15 steps. And it's not just necessarily a linear A to B to C to D to E to F. It can be, an A to B, but also A to B prime. So there are these forking networked pathways. And you can break different points in those pathways, which can affect the level of melanin that is synthesized and output into the animal. Then just completely aside from that whole melanin synthesis pathway, once melanin is synthesized, you have to pack it up in the melanosome and send that melanosome to where it belongs in the body. So you have a whole other pathway for how to make that melanosome. If something goes wrong in how that melanosome is put together, then you might be able to make all the melanin in the world, but pretend the melanosome is like a wiffle ball. The melanin goes into all the little tiny holes in the melanosome, and then it gets carried away. Well, what if you have a mutation where all the holes in the melanosome shrink down to the size of like a pinhole, but the melanin molecules are the size of a pea? Well, now you can't get them into the melanosome So no matter how much melanin you make, the melanosome stays empty. And then when it goes off to its destination, now you don't have any melanin at the destination because it all got left behind and broken down because it wasn't packaged away. And then you have another pathway that controls how the melanosomes get to their site. Well, if you break that pathway, it's like blowing up the highway. The highway's blown up, the car can't get to where it needs to go. So now you can break 3 different pathways, and each of those pathways can be broken up in their own networks for how they work. So that's how you can get different kinds of albino-looking animal. Well, only one of them's the true albino, and the other ones are, I don't want to say false albinos, but they're not albinos dealing with the melanism.
Got it.
Now, when you look at things like retics, where you have the purple albino and the white albino, when you look at ball pythons, you've got candy and albino. You have it in boa. I want to say it's the sharp albino and the boa woman caramel. Don't quote me on that. I'm not a boa person.
Okay.
These are compatible genes. If I put a white retic to a purple retic, I get a lavender retic. If I put a candy albino ball to an albino ball, I get the candino. If I put a sharp and a boa woman together, I get the paradigm. They're allelic combos. So it's the same gene that's mutated, but it's mutated in 2 different ways. And the way it works in those ones is one of the mutations is destroying the gene, basically. And that's what gives you your true albino. But the other one, it doesn't destroy the gene. It just renders the gene less functional than its normal. So now the gene still works, just not as good. So since it doesn't work as well, you get a little bit of pigmentation from it. And that's why, it's white and purple. That purple's getting a little bit of melanin, which is giving it that sort of purplish hue. The candy color, again, that sort of purplish, pinkish candy hue. With the boa woman caramel is I don't want to say a very colored animal, but compared to an albino, it's very colored. They call it a caramel because it's more caramel colored. And when you combine them, you get a toning down, because 2 copies of the partially functional is generating, X level of pigment, but only 1 copy is producing only half of that amount, so it can't get quite to the same color level that the 2 copies can.
So would it be accurate to say it's kind of like a suppression of the phenotype that's being expressed, kind of thing?
Yes and no.
Okay.
In terms of genetics, suppression means something completely different. Suppression is basically you're stopping something.
Right. Yeah.
The mutation isn't stopping it. It's more...
It's toning it down, kind of thing?
It's toning down. Let me use analogies. This is going back to communication. Just if you're trying to get a concept across, sometimes rather than talking in the big scientific jargon panel or way of doing it, it's easier to go by analogies, because analogies can be a lot simpler to understand. So, albino. With an albino, I have torn the wheels completely off of your car. Your car has, or the tires completely off of your car. Your car has no tires. That car is not going to drive well at all.
Yeah.
With the purple retic, with the candy ball python, with the caramel boa, I leave the tire on, but I've let all of the air out of the tire.
Okay.
It's the tire's there, but it still works like crap.
Yeah.
Now, when you have the combined to give you the lavender retic, the candino ball python, the paradigm boa, that's basically 2 of the wheels. I've taken the tire completely off, and 2 of the tires I've let the air completely out.
Got it.
So you
Okay.
get the
Yeah.
combination of completely non-functional and kind of sort of functional
Right.
versus 4 completely non-functional and 4 kind of sort of non-functional.
Okay. Yeah. Yeah, that makes perfect sense with bringing that car analogy in, because you don't want a car like that at all.
No, you
But
don't want a car like that.
right. Yeah. No, but it totally makes sense into like how those genes are functioning and really like working together in concert to produce, what we see visually, from, what's happening actually inside from a genetic standpoint, from what we can actually see as like herpers and things like that.
Yeah.
So yeah, it's super cool. Just going into kind of the deep, kind of the nitty gritty of the genetics, the things that you don't really see, because people talk about these things and kind of have done so many ways to really explain these things that are happening. Some that are accurate, some that are not accurate at all, and this is where you get this mess. But also just kind of breaking it down and adding analogies too helps me definitely, and I know helps a lot of people on the other end of it.
Yeah, and again, just in terms of being more literate about it, you can look into these things in terms of mutations in other animals. Our reptiles are not special in their own right and having weird mutations. You know, piebald. Everybody goes wild over piebald animals, thinking about how piebalds are awesome. But very few people recognize that those black spotted white rats that you're feeding to your piebald ball python, those are just piebald rats.
Yeah.
Nobody thinks that the piebald rat is all that awesome
Right.
because they don't stop and think that it's even a piebald rat. I'll go one further. If you are anywhere near a dairy farm, those black and white spotted cows, piebald.
Right.
Your Dalmatian dogs, piebald. So, the herp hobby doesn't exist in a vacuum when it comes to mutations. And so you can understand things a little better. If you broaden your sort of worldview, then you can broaden your reading. There are papers galore on piebaldism in rats, mice, hamsters, rabbits, cattle, dogs, cats.
Yeah. I think
It's
even when
out
we're
there.
just talking about mice, like just the genetics, like that's such a great way to start in reading papers on mice. And there's so much genetic information because they're tested so much within the science community, and then transferring that over to reptiles, you get so much great information from just doing that in itself.
Yeah, and then you can use that to it informs you about things. The recent release of the genetic testing that's going on, mostly for ball pythons right now, but I know that certain groups are looking at branching it out. But when these people were first starting, one of the things that I said when I heard about it was they were testing albino, and I mentioned it in conversation somewhere. I'll be interested to see how many different alleles they find for albino. And the person I was saying that to was like, "What are you talking about? That albino is albino." And I was like, "No, I bet that there's more than one mutation that still gives you an albino phenotype, and the phenotype is the same, but the gene will actually be broken in different ways." And like, what are you talking about? And I said, "Well, with mice, there are 12 documented mutations to that gene, and each one is different, but they all give the white mouse with red eyes. So
Yeah.
because I know that about mice, and it's the same thing with the albino zebrafish, with rats. We've already seen it in other places that there are multiple ways to break that gene and have it be completely turned off and broken. So it just made sense to me that we would see that with ball pythons. So then when the Ball Python Genetics Project released that they found 2 alleles of albinism in ball pythons, I was like, yeah, that
Makes
was
sense.
obvious. Yeah, that was
Yeah.
obvious to me. And I like I said, I'd had that conversation with someone. So I wasn't as I mean, I was excited to know that they had done it, but I wasn't as excited as like everybody else was with like everybody else just seemed to have their mind blown by that idea.
Yeah.
But, you know, if you go out and you read these papers on things and realize that your ball python that's an albino is not really different from your mouse or rat that's an albino, then these things, they may not come as a surprise to you, and it may then help you understand more, because once you've read that thing about albinism in mice, then you're like, okay, well, what other things in mice? Piebalds, okay, you know, what genes can cause piebaldism in mice and rats and dogs and cats and things? Well, there's only like 4 or 5 genes that do that. So now if we're trying to find piebaldism in ball pythons, we can look at 4 or 5 genes instead of looking at the 30,000 that are in there. And that's what one group did. And by taking 30,000 genes and making it 4 or 5, it's a lot easier to look at 4 or 5, and we now know the piebald gene in ball pythons. So that could be an easier way to help answer some of these questions rather than just going with the giant data crunch methodology. Now, some mutations will still have to just do that giant data crunch, because while there may be shared genetic mutations in other animals, the presentation may just be completely different because of the nature of those animals. You know, it's the reason an albino mouse looks different than an albino ball python. You know, albino mice don't have yellow spots on them because mammals only have melanin,
Right.
whereas reptiles have melanin and xanthin or erythrin, which essentially are the same pigment but processed different ways. So albinos in reptiles and in birds are a lot more colorful because they still have those red, yellow, orange pigments to them that mammals just don't have.
Yeah, exactly. And I think it's even really cool to compare different species. And when you're really digging deep into it, it like really just grabs your passion for reptiles even more, because you just understand how cool they are, not just from like a cool, pretty looking snake kind of thing, but also from a genetic and a biological and microbiology standpoint, that like these creatures have gone through so much through evolution and are able to do these amazing things. And it just blows your mind the more you get into it.
Yeah, it's that shared history and lineage through time.
Right.
It, you know, at least for me as a scientist, you know, it's somewhat humbling to know that, you know, in some capacity, the snake and I really aren't that much different, because a lot of the genes that are in me are in that snake.
Yeah.
And the same way that I share history with that snake, I share history with everything else on the planet. And, you know, I'm sure I've just made a whole bunch of creationists roll over in their grave. And I'm sorry, but this is one of those places where you can hold up a cat and tell me it's a dog, but I'm going to disagree with you there. And I think I have a pretty good substantial body of evidence to say otherwise. But
for anybody who wants to attack you, we'll put your email out there in big bold letters.
No, we can attack me. The creationists can say whatever they want. Just I'll tell you right now, I'm going to ignore you, because hey,
straight to spam.
Straight to spam.
Yeah, that's hilarious. Yeah, just one more thing I kind of wanted to talk to you about, specifically with leopard geckos and within the leopard gecko community, is just the snow gene. So we have the snow gene. A lot of other a lot of other different reptile species have snow. For most people, it's the anery and albino, but for us, it's just like you have your leopard gecko that's, you know, yellow with black spots if we're talking about the wild type, and the snow gene would just be the white background with the black spot. So we actually have 4 different types of snow, one which is incomplete dominant, and the other ones act in dominant faction. So the other 3 are dominant. You cross to a normal 50%, should be snow. So what's really interesting about the snows is they look exactly the same. So without knowing the history and the background of the parents, you wouldn't be able to tell which snow is different. But when you cross both snows, you kind of get the same effect as getting the incomplete dominant form, where you get the super form, the super snow that hatches out. But if you cross both dominant ones together, you won't get that, no matter how many times you cross them together. So do you think this is kind of an allelic thing happening, going on, and getting that interesting look where these all these animals look the same and genetically they act differently when you cross them different things, but when you cross them together, they produce what the super form of one of the snows would look like.
Um, okay, let me make sure I understand what we're talking about here, because leopard geckos are not my normal stomping grounds. Okay, so
Sure,
you've
sure.
got we have A, B, and C snow that you say are dominant. Now, if I breed two A snows together, do you get from that homozygous snow animals that look the same as the heterozygous snow animals?
No,
Like if I'll get a snow out of that breeding somewhere where if I
right.
breed it, all it throws are other snows.
No, it won't be. Yeah.
Okay. So what that is telling me then is that those three that you're saying are just simple dominant, they're also incomplete dominant, but you're looking at a super form that is lethal. Um,
Interesting.
Kind of like with the spider ball python. Um, Another example that for people who don't deal with ball pythons is probably a lot more recognizable is dwarfism in humans. That's traditionally labeled as a dominant gene because again that's artifactual from before we had the genetics to really test it out. So human achondroplastic dwarves have one normal gene and one dwarf gene. If two dwarf individuals breed together and have babies, all of their dwarf children are one normal gene, one dwarf gene, because if you get two dwarf genes together, that embryo dies before it can develop.
Okay.
So you are the fact that you don't get snows that breed as homozygous snows tells me that that homozygous phenotype is it doesn't exist because it's lethal. So you're
Okay.
never hatching them.
Interesting.
So all four then of your snow types are incomplete dominant. You just have three that have a lethal homozygous form.
Got it.
Um, so that would be the first part. Now, the second part, if I breed A and B together and I get an AB snow, basically, you're saying that that AB snow looks the same as the homozygous D snow, which is the one that's traditionally been considered to be incomplete dominant.
It would look different.
So the AB would look different than the super D,
Yeah, than the Super D. Yeah.
and the AC would look different than the super D, and the BC would look different than the super D.
Yeah, they'd all look different from that super.
Okay. Um, and then, but just in the broadest of terms, A looks like B, looks like C, and A, B, and C look like super D.
So they all look in their basic form. They all look this. They all look the same. And then
Okay, so
once
A
you get
looks like
that
B, looks like C, looks like D,
right,
but super D looks different,
yeah,
and AB, BC, and AC look different than super D.
yeah.
Okay. Um,
I wish we had visuals to.
I'm. It would help if we had visuals, but I'm able to put the pictures together in my head. So what then you're probably dealing with is, well, what you're obviously dealing with. You're getting mostly white animals, but they still have black pigmentation, just like the black spotting. Essentially, if you took a normal leopard gecko and pulled all of the yellow off of it,
Right. Yeah.
the patterning is the same as the adults would be, or as a normal would be.
Yeah.
Okay, so what you are probably seeing then is different forms of uh, basically axanthism. You're removing the yellow pigment from those animals. Now, I don't think it's as simple as just disrupting the pigment pathway. especially considering the fact that you have 3 of these that have a lethal superform, or that appear to have a lethal superform. So I would guess it's something that has to do with, when I was talking about the albino and how you have those melanosomes and how they develop and package, or the system that transports them. I'm guessing you're looking at systems like that that are disrupted, because usually those systems, they're involved in a lot more, like melanosomes are involved in a lot more than just holding melanin. Um, they're tied to the way, the way they travel is inherently tied into the way that the nerves lay down and travel through the body. So if you disrupt this pathway that causes melanin and melanosomes to travel, you're also breaking that pathway for how nerves form and develop through the body. So I'm guessing that you're probably looking at something like that, where if you're disrupting pigment cells, you could be disrupting on a broader scale. So the melanosomes, obviously you'll have xanthophores, which are the yellow packaged pigment cells. Those packaging cells are themselves basically the same. They started from a very similar precursor at the bottom, and then they branched out and went different ways on their pathway. If you disrupt that very basal pathway, now you mess up how all of those vesicles are formed. And while you have the vesicles that form the melanin and the vesicles that form the xanthophore, you also have vesicles that are important for cell signal processing, for immunologic responses, for cellular digestion, and things like that. So if you break that very basic bottom pathway, now you've disrupted a cell's complete and total ability to basically do anything. So those cells all die, and that would explain why you would have that lethal superform.
Got it.
Um,
I actually, let's see if I could get this share going. Maybe I could show you some of these. Can you see that right there?
Something's okay. Yes.
Yes. So this right here, that's the max no that you see.
Okay.
So that's going to be the incomplete dominant, or you get 2 max and then you could produce that super. This here is the tug snow, kind of pretty much looks the same. That's the dominant. The Albi snow, another version of the dominant.
So
Gym
I'm
snow.
still just only seeing the max snow right now.
Interesting. Okay. Let's see. There we go. How about there?
Okay, yeah, now I see the tug snow.
That's the tug. That's the dominant. It's the Albi right here.
Okay.
The gym is this one right here.
Mm-hmm.
And then here is that's super max no.
The super mac.
Yeah. So if you cross that mac to the tug, you could get an animal that visually looks like this. But if you get 2 tugs and breed them together, you won't get this.
You just get tugs.
Yeah, you'll get
normals.
all tugs and normals.
Tugs breed out tugs and normals, and none of them breed out super tugs.
Right, no. So you won't have like a 2 copy, so to speak, where you get 1 tug that produces all tugs.
Okay. Yeah, having that visual helps some. Now, I'm going to go back and look at some of these myself on the... So pardon the typing and me staring strangely into my computer screen.
No problem.
Yeah, so looking at these the gem, at least, and you can see it a bit in the tug too. As they age, it appears they get a slight bit of yellow pigmentation to them.
Mm-hmm.
So that would sort of lend to the idea that, again, you're disrupting something that deals with the ability of those pigment vessels that package and hold the yellow pigment. Um, so I would guess that, yeah, again, what you're probably looking at is different mutations that disrupt the formation of those vesicles
Got
or
it.
disrupt the way those vesicles are transported and moved around. And while a single disruption is enough to basically render them hypoerythristic or hypoxanthic to the point of looking black and white, having both copies of that gene mutant just ends up being lethal because you've disrupted a critical system, and the body cannot survive without at least one copy to help facilitate the motion or the packaging of those materials, not because it's important to the pigment, but because it's important to some other secondary process in the body.
Okay.
Um,
Yeah, and it's totally possible too. I know another big area of debate, like, is Tug, Albie, and Jim just all the same thing, and there's just different sects of that same thing because it's been called different things.
yeah, well, I mean, that would be very difficult. Like if you can breed a Tug to an Albie and not get a snow-looking animal that when you breed it, you get Tugs and Albies and normals, because and also Tug Albies.
Right.
You would need something that can get you all of that as one. All of that in one animal, versus just independent instances of the same gene.
Right. Exactly. And even then, it's like, how could you even tell? Because they look just so similar.
Right. And
just from test breeding.
That's kind of like, I hate just constantly bringing things back to ball pythons, but that's where I'm most versed.
Yeah.
So, like, in ball pythons, there are 5 different types of axanthic. Maybe it's 6 different types of axanthic at this point. I don't know. None of them are compatible with one another, but they all have a very similar phenotype. Now, those don't have a lethal superform because it's the homozygous form that you see the axanthism in. But if I breed a visual to a visual, I get double hets. If I breed those double hets together, how do I know that the axanthic that I got is a VPI axanthic or a TSK axanthic or a double VPI TSK?
Yeah.
Well, first I would have to take those axanthics and breed them to a VPI and a TSK. And if I breed it to a VPI and I get no visual VPIs, then I know, okay, that's not a VPI axanthic. If I breed it to a TSK and I get no TSKs, then I know it's not a TSK axanthic. If I breed it to both of them and get VPIs but no TSKs, then I know it's only VPI. If I breed it to both of them and get visuals from both of them, then I know that it's the double. But that's a lot of breeding work that has to go into it before I actually get my answer.
And who's going to go through all that pain in the butt to just to get that answer for that one animal?
Right. And I have gotten in this argument a few times in the ball python community where people like, it's not worth it because you'll never know. And my counter to that would be, well, you don't know that you wouldn't know, because maybe if you had the double visual, it would actually stand out as something completely different.
Right, true.
I don't know that, but I'll
There's
admit that I don't know that versus you just saying you'd never know.
Yeah, exactly. And that also interests me too, because it's like, what if that, brings a totally different phenotype depending on how those genes interact with each other, which is crazy to think about. Yeah. And there's all these other possibilities with different things and different test breedings, especially with ball pythons, just because leopard geckos, it's a little easier because just the time it takes from a hatchling to get to that breeding size. But with ball pythons, waiting those few years for that female to grow up and things like that. People just aren't willing to wait and put space to some of those experiments.
Yes. And I mean, even while it might be easier just in terms of timeline with leopard geckos, I would imagine that, well, yeah, each individual clutch is only 2 eggs.
Right. Yeah.
They clutch every 30 days.
Yeah. About.
So that's 2 eggs every 30 days. Over the course of a year or 2 years, that's a lot of little babies that you got to deal with and house.
True.
To then also raise up. Plus, if you're doing it to multiple, of these animals to try and sort this out,
Yeah.
you're suddenly up to 24 to 48 to 96.
Yeah.
It gets intimidating there after a while.
Yeah, definitely. Yeah. You will have a nice sample size, but.
Yeah, you'd have a great sample size, but at the same time, are you going to devote all of your breeding just to sorting that out, or are you going to devote your breeding to doing the things that you want to do? And, I'm not saying there's a right way or a wrong way to do it, but there is what you want to do for yourself. And if you have other projects that you want to work on, it's probably not worth it to you to just throw down and do this for the sake of answering the question for the hobby, because why the hell not? Then what are you going to do with all those babies that nobody wants because they were just part of a fun experiment to find out? Well, I'll just wholesale all of them, and then you get ripped apart because you decided to wholesale all of them. Actually, if I can segue off of that to go on a small rant,
floor's yours.
Because this just came up recently for me. In terms of breeding, I want to address and put it out there, and I will own that maybe this is just my feeling and my belief, but it's still something that I think kind of needs to be voiced. I feel like it's a little bit of a dirty, dark secret of this community, industry, hobby, what have you, that it's almost implied that unless you are breeding, you're not really part of this hobby. And that kind of bothers me, because in some respects, I think it's a very damning thing to do to new people getting into this hobby, to force them into it thinking they have to breed. There is absolutely nothing wrong with just being a keeper. And, I came into this hobby just being a keeper. Like I said, I bought that albino corn when I was 14 years old. I did not buy or keep another animal for nearly 20 years. And when I started getting other animals, it was more I'm an adult now, and I really like snakes and things, and I want to keep more than just one snake. It wasn't I've decided now that I've kept one snake for 20 years, I have to breed other snakes. So I just picked up singletons of things. The next couple snakes that I got were for, most of my life. I had the Audubon guide, and I fell in love with the gray-banded king snakes in there. And so I decided I wanted to get a gray band. I remember seeing that National Geographic cover with a chondro python just curled up in their quintessential way, and thinking, I want that snake. I don't know what caused me to go down the vein of blackhead pythons, but I remember seeing one of those and thinking, that's just awesome. If there's ever a chance for me to have one of those, I want that. Those 3 snakes, I dubbed my trinity, my holy trinity of snakes.
Nice.
So yeah, when I finally decided that I was going to start keeping more, and that I could do that because I was an adult now and I was only answerable to myself, the first thing I did was I found somebody who was selling gray banded king snakes, and I got a gray banded king snake, just one. And I still have her, and she's 15 years old. And 5 years ago, she had surgery to remove a tumor from her kidney. You know, she came in as a pet. She will always be a pet for me because I cared about how awesome the species was, and I didn't want to breed her. After her, I got a chondro. Now, she kind of fell into my lap because I ended up being friends with a guy who one day we were just talking snakes, and I had mentioned that, I was looking at maybe getting a chondro in the future, and he said, "Hey, I've got one that I'm not doing anything with. She's all yours." That animal is 20 this year, and she is just, again, a pet. She's got a nice big cage. She's up in my bedroom. I get to see her every day. These are pet animals for me. They're animals that I keep just for the sake of being a keeper. Now, yes, I admit I do breed, but that is more because, again, I'm a geneticist at heart. I'm a geneticist by trade. I'm my own personal mad scientist. And it's a lot better for me to be playing mad scientist with snakes than bringing my work home with me, because we don't want me to bring my work home with me.
Right.
But I while I breed my ball pythons, I'm not doing it for the, you breed because you can make money. You breed because you'll be the next big name. You'll breed because you make the next world's first and everybody thinks that you're awesome for making this combination. Like, I have made more world's firsts than I can keep track of, and I don't care, because my goal isn't to make the world's first. My goal is to make these things that I find interesting for myself. And if that's the kind of breeding you want to do, great. Have at it. But at the same time, if you just want to get into reptiles to get into reptiles, don't feel like you have to breed. You are absolutely just as legitimate as anybody else in this hobby if you just keep one leopard gecko. Or if you have one leopard gecko and one bearded dragon and one corn snake and one ball python. You don't have to breed anything to be a legitimate reptile person.
Yeah, I appreciate that, and really just breaking down that stigma, because it is a big stigma we have in the hobby, and I think that's coming even more when we're looking at reptile shows and people are getting like rack systems for their pets, and it's like, if you're not breeding, that kind of thing, it's not something that you have to get into to be a part of this hobby. There's so many different ways to be a part of it, and breeding is not the only way, definitely.
Yeah. And like I said, I went on a little bit of a rant there because I know it wasn't necessarily what we were going on with, but it was just, it had recently happened that I had a conversation with somebody about this. So it just thinking about, if you're breeding all of these different weird snows to try and find the answer, and I made the comment of, well, I'm just going to wholesale them all, and everybody thinks that you're a terrible person for wholesaling all those animals. You're not. If you do that just because it's a question that you want to answer for yourself, and you're not looking to sell them, and you're not looking to do anything other than answer your own question, that's great. Honestly,
especially if it's going to a good home, that's really pretty much all that matters.
Right. You don't have to be some huge seller and be awesome, and but I also don't want people to think that they have to breed either. If you go up to a table at a reptile show and you are interested in that leopard gecko that's sitting there because it's this beautiful black and white gecko, or it's this super orange gecko or whatever, and you pick it up and you're looking at it and you're in love with it, and then the guy behind the table is also like, and if you breed it with this one, and if you breed it with this one, you don't have to listen to him when he says that. If you find that one animal that you love, great. you can walk away with that one animal that you love. You don't have to walk away with two animals just because that guy is implying that the only way to be legitimate in this industry, in this hobby, is if you're a breeder.
Yeah, and it's interesting because I feel like I'm seeing that more, where more people are just spending a couple $100s on an animal they just love, and they set it up in a great home, have some beautiful decorations, set up their whole enclosure and really deck it out, and really just love that animal as if it was another cat or dog or something like that. And you see that happening a lot more. It's not like this disposable pet that they're getting. It's something that they're investing in and just getting it because they love it. And that's what we all do. No one gets, well, I guess some people now, but like as you're a kid, running around catching stuff in your backyard, you didn't get into reptiles because you're like, whoa, look at this thing that I could breed in the future possibly and make all this money. It's like you're genuinely fascinated and have this heart and this animal that you love to see, and you're just fascinated and curious, like, why does it have scales, why does it not have skin, kind of thing. And that's what gets us all in the hobby in the beginning.
Yeah, and when we're keeping them as kids and stuff, we're always putting them in the glass tank with the grass from outside and the sticks and we as kids we're interested in doing it to have that little piece of nature
Right.
in your house with you. And I do appreciate now that we're starting to see a little bit of a shift in the hobby where, I think the reptile hobby as a whole would be so much better if we started adapting the mentality of like fish breeders, where, like you said, they just buy a, $100-200 pet animal, but then they're spending $300 on a cage and
Right.
$200 on a thermostat system and $150 on lighting and heating and
Filtration, just the whole thing.
filtrations. And like you buy, you think of the fish hobby, they buy a $5 fish and they put it in $1,000 aquarium with all the kit and stuff, like, and that is seen as legitimate in the fish hobby. I think we need to see that as legitimate in the reptile hobby too, just as legitimate as the guys who are breeding. I'm not saying that one is better than the other. I'm saying you don't have to breed to be legitimate. I don't think you have to go all out and go crazy with cages and stuff to be legitimate, but I do think that moving towards that, especially for newer people, is a good thing because it keeps that passion, and it goes back to really how a lot of us came into it, with that fascination with nature and that wanting to have a piece of nature in your house and appreciating these animals, not just for what they can give you by breeding them, but just for what they are as these little monsters that we keep.
Right, exactly. And you get to see a little bit of yourself in that kid that's interested in the same way that you were. And it's cool to also just share that passion with the next generation, which we see happening already, which is also awesome.
Yep.
Nice. Well, again, I'm just so grateful for this conversation, and I'm glad you're able to come onto the podcast and really just share all your knowledge and give us a lot of great advice with genetics and so many other things today. So just thanks again for coming onto the podcast.
No, I appreciate you having me on again. Thank you very much.
Definitely. So at the end of each episode, we kind of do a few fast round questions to just close it off. So is that cool if we do a few fast round questions with you?
Yep, sure, shoot.
Cool, nice. So are you a early bird or a night owl?
I'm probably more early bird than night owl.
Okay. Coffee or tea?
Tea. Absolutely tea.
Nice. Do you have a favorite tea?
Tea. I really like chais.
Okay.
So
nice.
I do a lot of loose leaf blends and make my own up, but chai, just a really nice spicy chai tea.
Nice. Sweet. iPhone or Android?
iPhone.
Okay. Computer or tablet?
Computer.
Books or TV?
Books.
Science or art?
Well, obviously I'm going to say science, but if I were really being honest, I think both are almost intrinsically tied to one another. I took so many art classes all the way up into college. I have an art portfolio upstairs just filled with drawings and work and stuff that I've done. I
Nice.
haven't had as much time to do it, as I have grown older, but I do believe that having that artistic, you know, I, I'm not going to claim that I'm, anything close to the artists that we have in the hobby, but at the same time, having that artistic outlet and that way of viewing the world is very beneficial. And being able to look at things in the abstract as well as just the logic and breakdown of science, I think is important.
Nice. When it comes to genetics, are you dominant guy or recessive?
I am recessive.
Nice. Okay. Do you have a favorite gene that you like out of all any reptile?
I'm very, very big into albinos. So
Nice.
albino was my first love. Like I said, I bought that AML corn, and I have always had some kind of albino, just because I really like, I really like those red eyes, that sharp contrast that you get. Uh, I work with albino and candino in the ball pythons,
Hmm.
and they're allelic. I
Right.
I like that little bit of pinky tone that the candino adds to it, but if I had to get rid of everything but one morph, it would be albinos.
Nice. Okay. I also know that you bake. So, what is your favorite thing to bake?
Oh God, that's just not fair. um,
If you're going to a party, you're cooking for the president or something. What are
if
you
I am
bringing?
going to a party and I'm cooking, the the thing that has been most popular when I've gone to parties and stuff, when I've gone to Northeast Carpet Fest and things, has been the red wine rosemary pear pie. Um,
That sounds amazing.
I I really enjoy that. Everybody else seems to really enjoy it, but yeah, I guess that would have to be probably my top pick. But I mean, I'm always baking new things because it's just fun for me. It's another one of those mad scientist things.
Yeah.
It's science that I can do at home,
Yeah. And you could eat.
and you can eat. Well,
Yeah.
I strangely I don't eat a lot of my creations, mostly because if
Interesting.
I did, I'd be like 400 pounds. um, that's it for people who follow me. They know every week I post a baking for the coworkers. That started like 7 years ago.
Wow.
I just I my kid was asking me to bake something for her class, and I needed to get rid of the leftovers because like everything that I made for her as an experiment she didn't like. And then I realized I'm having a lot of fun with this, and so I
Yeah.
just started doing it every weekend. I would bake something and take it in on a Monday, and so it just became this thing for me. And
Wow.
it's it's a lot of fun to every weekend come up with something new and different to bring in and share with my coworkers, and I don't have to eat it, so it's not sitting around here making me 400 pounds, and
Right.
I'm bringing a little bit of joy to the workforce.
Nice. Awesome. Well, again, I appreciate you for coming on to the podcast and yeah, just sharing all your knowledge with us today.
Thank you again.
Awesome. See you next time.
Catch you later.
And that's it for this episode of the podcast. I really appreciate Dr. Travis Wyman for coming on to the podcast, really sharing so much information, and not just coming onto the podcast, but all that he does for the hobby. He's been on so many other amazing podcasts that have to do with reptiles. He's written articles, even doing his own research. He's just contributed so much to just what the hobby is, really just getting that information out there, spreading that information, and using his education to really educate the broader reptile hobby about genetics and so many other things when it comes to genes. And again, he just does so much for the hobby. So I really appreciate all that he does, not just for coming onto this podcast, but everything else he's done for the hobby. As you guys could tell, he has so much passion for the hobby, and that really just goes out to the information that he shares, the hot takes that he has. I don't even know if they're hot takes, but just really great information and the passion that he has for the hobby and sharing that with all of us today. So I hope you guys really appreciated this episode talking about genetics and the things that you might not know about genetics, and then also bringing in Leopard Gecko genetics into that. As you guys know, at the end of each and every episode, we do our one big thing. And for this episode, I really want to just talk about getting information out there to people about genetics. There's so much stuff out there in the hobby, and it's so hard to sift through information and finding out the right terms for genetics, how Mendelian genetics interact with each other, how certain heritability gets into each other, and how it's passed down from generation to generation, especially when it comes to leopard geckos, right? We have base morphs and genes, and we also have line bred things. It's so confusing, especially when you're first getting into the hobby. But really just looking into other research, I really implore you guys to go out and look at other research and read other papers. Go and listen to other talks done by researchers like Dr. Travis Wyman, Warren Booth, so many other people. We have an awesome episode with Dr. Ben Morrill, who's been on the podcast before. Make sure to go back and continue to do your own research and look into the interest when it comes to genetics. Go look at papers that we talked about mice, about other species that can be correlated to leopard geckos, and really understanding how these genetics work and how we could get this information and apply these things within the broader area of biology and bring it into leopard geckos, and making sure that we're all on the same terms when we're talking about genetics, but also making sure that we're competent as a hobby talking about genetics. So that's my one big thing for this episode of the podcast. I hope that you guys really appreciate it and can continue to share this knowledge that we talked about today on the podcast. Thanks again for watching the podcast. I hope that you share with a friend, rate the podcast, and make sure that you subscribe. Make sure to listen on all the podcast platforms that we have, Apple Podcasts, Spotify, Google Music, and also YouTube if you guys are watching this online. Make sure to check in next time for our next episode on all those podcast forms that I mentioned before. This is your host, Evan Wooldridge. Continue to grow in knowledge and share the strength in Leos.